Welcome, everybody. Thank you for joining us. Thank you for joining this ClawBio and Genomic Intelligence hackathon. I'm going to run the first 20 minutes. And in these first 20 minutes, I will be providing a little bit of the framing. Then Benjamin and the Genomics Intelligence team take over with the live work. One line to hold on to for the whole talk. Agents can now write and do a lot of work. They can run the analysis, they can open questions, they can do a lot of work. But the open question is whether you can trust what they run. Everything I show is built around that single problem. Quick orientation. So I take 20 first minutes to set up the problem and show you the evidence. Then Benjamin and the Genomic Intelligence group will show you the models and run live scenarios inside ClawBio. We close with Q&A and an open call. Build something on the platform over the next 4 nights and show us. Move fast through this. The audience just needs to know where the handoff is. Two waves. The first wave of LLMs in biology was retrieval. We summarize this paper, answer a pathway question, pull structured data out of text. That was genuinely useful, but incremental. It didn't change how we work, only how fast we read. The second wave is different in kind. Moderns model write, debug, execute code, and when you connect with them to a file system, databases and command line tools, they plan multi-step analysis and adapt as intermediate results come back. So the consequence is that the thing to hold on to, your job, shifts from producing the analysis to judging it. Recent reviews catalog dozens of these systems across genomics, single-cell proteomics, and this is not one lab's claim. It is a field-wide move, and we are just at the beginning of it. Let me then define what we mean by agentic genomics. 4 conditions jointly necessary. The agent decides at runtime. It works through a constrained library of validated skills, not code invented on the spot. It diagnoses its own errors and replans, and it is directed in natural language. If a system only satisfies some of this, then it's something else. It can be a workflow automation, AutoML, or a chatbot. The clean discriminator is the perturbation tests at the bottom. So change an intermediate result and watch. A real agent replans, and a fixed directed acyclic graph carries on regardless. So let me give you a concrete example. You have BWA Ven2 that throws an incompatible index error in mid-run, and the agent diagnoses it, rebuilds the index, and resumes. No human in the loop. That self-repair is the property, not the speed. So I had to compress the whole perspective into one sentence. And if I was to do so, then this is it. The hard rate limiting step used to be building the pipeline. Now, agents have largely dissolved that. What they have not dissolved, what they actually make harder, is knowing the result is correct. So the rest of this talk is about that gap, how it fails, what it costs, and what we do about it. Why is this validation the hard part? Because of how agents fail. So we have 3 properties, left to right. The output is plausible, well-formatted, fluent, exactly what you expect to see. It's confidence. A wrong answer delivered in the same tone as a right one. No hedge, no flag. And in our domain, it's consequential. The failure mode isn't a broken web page. It's a missed cancer gene or a wrong drug dose. The button at the box is labeled as illustrative deliberately. It is a constructed scenario, not a clean case that I observed. But imagine a BRCA1 frame shift, which is quietly downgraded to likely benign. And you get a clean report, real-looking citation, no flag raised, no screening offered, nothing crashes. Now imagine that pipeline run across 10,000 genomes overnight. The next slide is not illustrative. It's actually what happened. So a ClawBio pharmacogenomics skill was handed an empty file, literally no genomic data. Instead of refusing, it returned a tidy, all normal report, including dosing recommendations for 51 drugs. No crash, no warning, and this is the silent failure mode in the flesh. What matters is what happened next. In the first weeks of our release, a computational biologist was stress testing the library in the open, found it, and disclosed it publicly. Not a quiet patch. In fact, this person told me that ClawBio could kill people. So this is not a trivial example. The lesson isn't about one or skill. It's that the governance loop, the public audit, and allowed fix is a first-class part of validation. Closed development of ClawBio or any of those skills would have hidden this. So open development caught it. And here is ClawBio. What is ClawBio? It's an open agent-native skill library for bioinformatics. It's open source, local first, reproducible. I show you some of the numbers here, but I want to be honest about the ones that mean something. Stars and forks are attention, not use. I put them in gray for a reason. The signal I actually care about is the middle line. Within weeks of release, wet lab biologists and with little code experience were contributing tested working skills, using the model as programming intermediary. Last week, I had 5,000 downloads of clones of ClawBio. I'm not claiming that I have 5,000 people downloading it, but certainly, maybe 1,000 would have downloaded those clones. And the interesting thing about ClawBio is that it only started at the end of February. So in 4 months has become de facto the open source community that encompass biological biodomain skills for agentic AI. So I think the point of ClawBio is that it's open, it's transparent, so the code is there, everyone can see what's inside, and it's auditable on openness. And this matters before Benjamin speaks, because genomic intelligence models use plug as in skills inside this library. What's open and auditable is the contract and the execution trace. So the trust story lives here around whatever the model sits in the middle. Does a plain text skill actually buy you anything? Here's the evidence, and I want to be precise because this number gets misread. We took pharmacogenomics deliberately because it's one corner of genomics with an authoritative ground truth. It's called CPIC guidelines, which is an international database currently accepted by major medical institutions that tells you specifically the relationships between gene and a drug. So we ran 44,000 scored evaluations across 9 frontier models, including O3, O4, GPT 5.2, Sonnet 4, Opus 4, and we scored these 44,000 evaluations in 3 conditions. Free prompt with no specification, then with skill reasoning, the stuff that you would have for a skill that you have actually put on an MD file, and then skill execution, where you actually give them a lookup table. And so what we find is that give the model the guideline as reasoning context, and then you get 95% of the time correct answer. Have the skill execute the guideline deterministically, you get 100%. Now, the honest reading of that 100% of correct answers in these thousands of evaluations is not the model becoming clever. It is the model taking out of the answer. The deterministic skill applies the logic of the gold standard database, not by asking nicely. That distinction is the whole thesis in one slide. In other words, if you want to get deterministic answers, then you have to take out the reasoning from the model. And if you provide as a skill, rather than the text for instructions, but you provide code that is executed, then you maximize the amount of correct answers that you get in an otherwise stochastic set of answers that you would get like from free prompt. So this is one way of improving that need for validation that we would require whenever we want to implement agentic genomics at scale as part of an infrastructure. So here's the result that should give everyone pause. And it's the one kind of thing we only found because we scored at scale. So when you give the model the correct guideline as context, the average accuracy goes up. The headline improves, most cases get better. But hidden underneath the average, a minority of answers that were previously safe flipped to confidence were wrong. And adding the right information created a new failure mode. And the average concealed it. This is why I don't trust prompt level fixes for consequential work. It's better prompt that can raise the mean and quietly introduce new, fluent, hard to catch errors. So trust has to be architectural, deterministic, auditable, the same regardless of which model is underneath. And something that is quite interesting is that when we provide skills as code that can be executed, you get 100% correct answers regardless of the model. So you might use, for instance, let's say Google Gemini Flash 2.5 or Opus 4, and you get no difference in performance when you have the model just orchestrating the running of the skill. So this is something that it caught my attention. Is something which is really useful for us to know, because it's not the same running a smaller model, which is a lot cheaper than perhaps one of the biggest or most expensive ones like Opus. And so this is a good consideration to take into account. Also, something quite interesting, we find that we got different results when we allow the model to reason. And these results, when the model reasons, actually get worse the further you are from the European ancestry. So in other words, the biases that we knew that existed in genomics, whereby you have underrepresentation of non-Europeans, we observe an amplification of that. And we tested it with individuals from East African ancestry, from the Uganda Genome Resource, and the Peruvian Genome Project of Latin American ancestry and Europeans. So an agent left to its defaults reaches the most abundant European resources and silently inherits this gradient at scale. But when the skill executed the guideline deterministically, every ancestry goes back to roughly 100%. So the fix for the equity gap and the fix for the trust gap are the same fix. And that's why I say equity has to be engineered, not declared. and it's a systems requirement, and therefore it is auditable. So I wish, you know, to talk about this particular aspect. What do we do about this? So let's not abandon agents. Calibrate scrutiny to consequence. And we have three tiers. The research grade, where you have exploration, where you need unit tests, adversarial inputs. We have another higher level, which is what I call benchmarked, where you really want to publish some particular work. You are much more stringent in the number of errors that you allow any agent to produce. You would need validation with public reference datasets, such as genome in a bottle, metrics with confidence intervals, and documented failure modes. And then we have the clinical grade, where we would want to use agents in a patient data setting. And the bar for the implementation of agentic genomics at that level must be 100% accuracy. How do we do that? Well, I think I've shown here that providing skills, not the ones that allow or permit reasoning within it, that the ones that execute code, and where you get the agent level at the orchestration and not the reasoning, that's where it seems that we could close that gap in terms of having results that are trustworthy enough for clinical application. That's not to say that I'm claiming in any way that this suggests that this is potentially the way. Well, I'm not claiming that this is the case. I'm saying that certainly this seems to be an avenue for this kind of validation that we would require at the clinical level. If we wanted to have hospitals using this kind of infrastructure at an infrastructure level. So just to conclude, before we go into my colleagues, I'd like you to remember three things, okay? One is that the bottleneck has moved. Agents produce faster than we can check. So the scarce, valuable skill is now judgment, not code. Two, trust is architectural. It comes from deterministic, auditable, model invariant execution, and not from a cleverer prompt or a bigger model. Yeah? Not from a cleverer prompt or a bigger model. It comes from architecture. And three, equity is engineered, not declared. So the accuracy gap across ancestry is a correctness gap. And the fix is the same fix as for trust. And so underneath of all these three sits one thing that doesn't automate. And the thing that doesn't automate is domain expertise. And that's the irreducible human contribution. You can outsource thinking, but you cannot outsource understanding. And with that, let me hand over to my colleagues at Genomic Intelligence. So over to you, Ben.
Thank you, Manuel. So, I'm Ben from Genomic Intelligence. And what is Genomic Intelligence? We are the company that builds AIware to understand genome and to predict all different kinds of genomic signals and function from the nucleotides. So it can be lots of different signals encoded in the DNA, and we have different models for them. And in this hands-on, I'm going to show how to use these models. And first, I will explain what signals you can extract from the DNA sequence with these models. Then I'm going to show you how to do this with various approaches, using the web platform that we developed, using the MCP server, like with your favorite AI chatbot. But of course, I totally agree with what Manuel said, that the most controlled and the simplest way to integrate this into your research or development is to use ClawBio skills and agents, and this will be the main part of the hands-on. So, couple of things to prepare. First, there is a link to a Google Doc where I prepared some useful links, instructions, and some other materials. I'm going to post these into the chat. Let me see. Yeah, you should have it in the chat if it's not convenient to you to scan this QR code. Also, I suggest that you open your favorite AI provider. I'm going to use ChatGPT and later on Codex. And unfortunately, the use of MCP server and Codex on your PC is possible only with paid subscription, but you also can use Cloud Code or any other system with the skills and with ClawBio. So you are not limited. I'm going to show it this way, but you are not limited to this. You can try in parallel all other types of AI agents. And yeah, I actually understand that we are all from different backgrounds. Probably many of you are from bio and some from IT, and I will try to balance between the level of detalization and the speed because I want to show a couple of examples. But if it will be a bit complicated to follow because of technical reasons or maybe because we move too fast, we published a series of posts, and you will find the links to these posts in a Google Doc that I shared. And feel free to read them and to follow instructions from these posts if it's easier for you. But actually, please experiment with this, experiment with prompts, experiment with our models, experiment with applications. Maybe even during this presentation, you can develop something because it's now so easy to use these models for some bioresearch. And if you have anything interesting in mind and you are able to create some applications right now or within the 2 weeks from this webinar, we have a capacity to support the cases that we love with a price of $1,000. So please write a short blog post describing what you have built and tested and send it to me. It's my email address here. It will also appear in the end of the presentation. And I'm super excited because I think there are so many applications now, much more than a couple of examples that I'm going to present. And another point also that was said, we have a chat here in Zoom, so please feel free to ask questions, to propose something, to engage in discussions. And my colleagues from Genomic Intelligence will try to answer right now, or maybe we can design something during this webinar. Okay, let's get started. So first of all, I want to share with you the models we have. And I think the easiest way to explore them is through the web platform where you can see inputs and outputs. Here is a link to the web platform. I'm going to follow it, but also you can see this link in a doc that I shared. So the first link here is to the doc itself, probably useless if you have already opened it. Then the second one is to the platform. Please follow it right now. So you can click here anywhere and just explore how it works. I will go through the models. So the one model is a classification model that can say, given the DNA input, which segments of this input has a promoter activity. So for those who are not familiar with the concept, the promoter is a sequence nearby the gene start, which determines whether actually this is a gene or not. So whether it will encode RNA molecule, whether it will be active or not. And if it's active, it determines in which cell types specifically this gene will be active. So let's have an example. You can provide the sequence as a text here in a box, but I would prefer to fetch some sequence around a known gene. We can fetch from ensemble with this balance or from the human genome with this one. Yeah, let's use this one, for example, and it will just fill the input. And also, yeah, you can upload, if it's a longer one, you can upload here the FASTA file. It's a very simple standard biological format, basically just nucleotides and the header, and use this option to provide the sequence. The sequence is the main input of all our models. So let's have a look what happens after we run analysis. The model will analyze the region we provided, and it will tell us that there are three segments which look like promoters. So from our biological grounds, we probably suggest that these three, they are segmented just because the model analyzed them by chunks, but probably this is one longer promoter region, and this is another one alternative promoter, which is quite common for genes to have several alternative starts. Then below, you can see different metrics about how confident is the model in each case. For example, these calls are very confident with more than 90% confidence. And here, for example, we should consider there is no promoter because the confidence is very low. There are also some statistics about distribution of this confidence. Probably doesn't make much sense for such a small region, but if you submit a longer one, you would like to see these distributions and some other statistics that you can explore. Okay, so this was about the beginning of the gene, but also you might think about how the gene itself is organized. And I will show later on in a slide, and we will have a specific use case for this problem, but briefly, the genes in eukaryotic cells, they are organized in exons and introns. So introns do not typically participate into the mature RNA or partially participate in case of alternative splicing. And there are signals which are called splice-sites, and these signals cause excision of these intronic sequences from the gene. So let's have a look how the model predicts the distribution of these splice sites. Let's take one of the famous SMN genes. They're actually famous because they are associated with monogenic disease. Not long time ago was treated with a very high efficiency, one of the rare and very successful examples of monogenic disease treatment. When we submit the sequence of this gene, we see that the model predicts where are those boundaries between exons and introns. So they are called like splice acceptors and splice donors, meaning that these acceptors indicate the start of the exon, the part that will remain in a mature RNA, and donors like indicate the end of this sequence. So every region between acceptor and donor, the smaller region, will stay within the RNA structure. And if you zoom in, you can see how this maps back to the original sequence. Like here, for example, we see that this small segment is actually retained in RNA after introns are removed. Okay, so another type of the problem, if we know where is the gene and how gene is structured, is to predict what is the level of expression for this gene. Expression means activity, like how active is this gene in different tissues. And since I'm saying in different tissues, that definitely means that to solve this problem, you would need to provide a context. You would need to say what cell types you are talking about. Is it hepatocytes, for example, or are these blood cells? Because they're quite different, and most of genes will have different activity between them. So let's, for example, select a hematoglobulin gene. So this gene encodes the globin, and it's highly active in blood cells. And let's actually use one example of such cells and run the assay. So you see, together with the DNA now, we have a text description of what type of cell we are considering. And this type of cell is very relevant to the task because if we change it, the results also should get changed. So what happens next? The model would say that there is one transcript, like one instance of this gene here, and it is important to use this model that its start should be located right in the middle of the sequence. And I will show you later on that ClawBio makes it very easy because it knows by instructions and skills how this should be located, and you don't need to think about it. But for now, in this example, we already did it all right, and we have this gene located, this gene start located right in the middle of the sequence. And yeah, we see here that there is certain level of expression for this gene, which is not very high. And we also can play like changing the gene and changing the cell type. For example, let's take albumin in hepatocytes and see the albumin is a classic example of hepatocyte-specific gene, and we can run again this assay, and we can see that now, for example, the model predicts a very high level of expression. And of course, we can combine, like if we have the models that can find where gene starts or find complete sequence of the gene, like the next model, this one for gene finding, and we have a model that can predict how active it is in different cell contacts, we can combine these models and run analysis where first we will search for genes in a sequence that we have provided. And second, for each gene that was found, we will estimate how active it is in a specified cell type. This will take a bit longer because we need now to make several rounds of predictions. And yeah, here, for example, there is no gene in this sequence, but we can switch to another region to like try to find genes and see how it works there. I need to say that those models that I'm showing now, they were trained using mostly vertebrates data. So probably they will generalize across lots of species in this taxon, but not far away from the vertebrates. But also we have dedicated models for cancers in fly. So this model was trained using Drosophila data, and this one can be used to predict cancer activity. So here, for example, before we used some regions centered around gene, and here I would like to fetch a region that includes some intergenic sequence and put it here, and let's see how it works then. So this model can output two types of signals. One highlights the developmental gene enhancers. So enhancers are kind of sequences that allow gene to be active or increase its activity. And these enhancers, they are specific to genes that are active during the embryo development. And another signal is activity for housekeeping genes. So those genes are active in most cell types both during development and during in adult animals. So you have these two types of signals here. And let's extend this example. So I briefly showed you different models and how to use them in a web platform. But of course, if you have a large batch of sequences, it will be time consuming to paste every time the sequence here in a web interface. So we really need instruments that can incorporate this analysis into the workflows at scale. And so now I'm going to show you first how to do this in a chatbot. While we are using an MCP server that we developed, and then we will move to agents with skills. But before I leave this page, I want to show you very important button here. So this button is a feedback where you can write anything about bug that you find, or maybe you would like to see more models here than we have currently available, and it will be very interesting for us to see what is the demand and whether we can supply with these models. So basically, any feedback you would like, please put it here, and we will try to react on it. Okay, let's now move on to another type of interaction between our models and how you can use them. And for this, we will need a ChatGPT or another agentic system. And again, I'm showing you specifically how to set up this in ChatGPT. There, you use the special apps button. But for example, for Claude Code, you would need to modify the config with these simple lines. And in general, if you want to learn more about how to set up this in a doc that I have showed you, here we have a link to setup page, and please read it. And there are several examples and highlights there. But for GPT, you need to go to this to this apps button that will end up on this apps page, and then you go to this corner and view the apps that you have. By default, you have many of them, so you need to add one more. For adding these, you will need a developmental dev mode accepted. So, and when you add more, when you push this add more button, you will then, let me refresh the page so we can see it. Let me see. Okay, so basically this is a screen you should have when you add more, and then you provide this URL. You can find it in a doc here. I put it specifically for GPT in a doc, and also for other systems, please read this page. And that's basically it. So after you enable this new app, it should appear in your apps under the more. You should have this Genomic Intelligence app connected. And once you have it, you should be able to prompt like something like this. So let's discuss this prompt. So what I'm typing here is like fetch sequence around Drosophila locus. There's a specific locus I'm interested in, and predict developmental and housekeeping and cancer activity. So let's see how it will work. After I prompt it, the GPT itself will decide which tools or apps to use. So by the prompt, it will understand that it should use the fetch option also provided in our MCP. Once it got the sequence from the locus, so the locus coordinates, I never provided it myself, but because the name of the gene was provided, it was able to find out the coordinates and get the DNA sequence. So then it will communicate with the model. Here it goes, this communication, and retrieve those results that I showed you using the web platform, but now you can have them embedded into your chart all together with some other information. Yeah, so you see it here. Like it found some strong and cancer activity similar to the web platform, but it also can summarize for you different parts of sequences and provide some meta information about results. So this is already useful because you don't need to manually find the sequence and do some other parts of this workflow. But still, it's hard to scale because this online version doesn't have access to the data you have. And as I said, the most efficient and the most productive way of doing this would be to use ClawBio and skills that are embedded there. So let's try to do this. And for this, we will need the ClawBio repo itself. And again, I'm going to show it in a Codex that will be embedded into the cursor environment. So first, to start Codex, we need to create a project, and we will create a project with opening ClawBio repository. You have a link in this master document. You go to the URLs, and here is ClawBio repo. So you just copy it and put it here. I will now wait a little bit, so you provide a name. Let's say something like this. I want it to be a new folder, so we can start from scratch. And I will wait a little bit, so if you are reproducing it right now in parallel with me, you have time to do the same. If you have Codex, please sign in if you need, because later on we will use it. So cloning of the repo is in progress now. Keep this document opened, and while it's running, I want to explain you actually what will be going on under the hood when we do this analysis with ClawBio, but actually when we do this in MCP and other way contacting to our platform. So what is actually happening under the hood, there is a code executed, and for analysis of the sequence, this code is using API access to our model. And API, if you're a developer, you might be interested in exploring this API. So the links that explain in details the API we have are again in this master doc under the API tab. But also, it's quite simple. So you just post requests to this address And these posts, they explain like what action and what analysis you need from the model and what inputs you have. So basically, they recapitulate what you see in a platform, but this is a way of automating this access, so you don't need to do this manually. Okay, so let's open now the repo is cloned. Let's open it. If it's a bit too fast, you can write in the chat and ask for support. I will move on with examples now. Let's open Codex here. And let's consider our first problem. So our first problem will be the genome annotation. That is quite common problem in biology. We have now a sequencing costs that goes dramatically down, and it's very cheap to sequence. Plus, we have fantastic technologies such as nanopore sequencing or PacBio sequencing, where you can have long reads, you have high-C, so you have so many tools to sequence and assemble genomes, but the annotation of these genomes becoming bottleneck. What I mean under annotation, like almost every biological analysis would start from where are the genes and what these genes encode. Do they encode proteins or they are non-coding RNA genes? And what is the function of these proteins? Like without these, those genomes value, I don't know, 10% of them. And if you will go to NCBI, which is the largest database for the genomes, you will see that the number of sequenced and annotated genomes is more than 10 times different, more than 100 times different, actually. And that's because we don't have good computational tools that allow genome annotation, I mean, finding gene, explaining what it encodes, and explaining what is its internal structure. I already described it before, like where are those parts that will remain in a mature RNA, and where are those parts which encode protein, if this is a protein coding gene. Yeah, so we don't have tools that find genes and understand their internal structure. That's a huge problem. We can do this annotation with some additional experiments, such as RNA sequencing, but purely from DNA sequence that is very accessible, we can do it efficiently. So let's see if we can solve this problem with genomic intelligence models embedded into ClawBio. So our prompt will be to analyze a region of human genome. We'll use human genome in this example because I want to validate how the model works. Of course, in a real-world task, we will probably apply this to some unknown species for which we don't have such a comprehensive annotation as for human. But right now, I want kind of ground true in this assay. So we will focus on just some random region on chromosome 8. And so the workflow, which I suggest, will be to get the sequence from this region, then to run Genomic Intelligence gene finding or annotation tool to identify possible transcripts or genes in this region, then to write Genomic Intelligence splicing tool to annotate potential splice donors and acceptor sites. You remember I showed this, so these boundaries will be splice donors and here acceptors. So if we know them, we can say what is internal structure of the protein. So once we have this, let's overlap the predicted boundaries, the predicted splice sites, with predicted transcripts and infer the likely exon and intron structure. Then, based on what we predicted for exon structure, let's identify if there could be a protein sequence encoded inside this region. Finally, if we get some candidates of protein sequences, let's fetch the sequence of a protein and let's blast the predicted protein sequence, like search the predicted protein sequence against the large repository of known proteins. And then in this prompt, I will give details. It's what Manuel said. So I might not give all these details. For example, I might not say that we want to use genomic intelligence model. I might not say that you need to read the relevant skills file, but I want it to be reproducible. So that's why I do it so detailed here. But again, you actually can experiment with less detailed prompts and see how they work. In my experience on these tasks, they work quite well without much details. Note that to access models, you need the API key. The API key is already embedded into the repository, ClawBio repository that you cloned, so you don't need to do anything, but it has a limited API call rate. So if you submit too many tasks or if your task needs to call two models too often, you may hit the upper limit of this API key, and you have this feedback field in our platform. You can submit an inquiry to get the API key with larger capacities, or you can write us. I have shown email, and there is my email in this master doc. And yeah, you can write me to ask for larger capacities key. Yeah, let's give it a try. So of course, I'm not coding anything. I will just put this prompt into the Codex system and submit it. And let's see how it will work. Yeah. So this actually, in my experience, will take some time. So we will start this process now. And yeah, you see, so it's following the guidelines to read skills.
We cannot see, Benji, we cannot see the Codex screen. We see web browser.
I see. Thank you very much. Just a second. Can you see the Codex now?
Yes.
Yeah, so let me switch the screen once again, and I want to show, because probably you haven't seen how I did clone the repo and open Codex, so I will show it once again in a new window. This one is already working, but for those who want to reproduce, so what I did, I just opened a fresh window, then I pushed this clone repo, then into this I paste the ClawBio address from the file that you have already. Let me show. Here in URLs, you would need to copy this ClawBio address, put it here. Okay, I'm not going to create another folder. I already have 10 of them, I think. But you need to proceed. And after you are done, I'm going to switch to the window I already have where I'll clone the repo. So after you proceed, you should end up with this file tree showing ClawBio structure. You don't need to go deep into this file structure. The agents will do everything for you. You just need to ensure that you cloned it successfully and opened the project. Then you need to open Codex or whatever agentic system you have. So for Codex, I'm opening the command palette, and here is an option, like you can start typing it if it's not in a top, Codex, open Codex sidebar. And after you push this open Codex sidebar, you should end up in the same state where I am right now. So I will briefly go through what's going on in this process, and you have like 1-2 minutes to replicate these steps. But just a reminder, if you are lost, no worries. Just follow the links on blog posts, and they explain exactly those steps so you can reproduce. Okay, so what's going on here? I think what an important part. So the system actually follows exactly the workflow that we have in the prompt. But what is important that it has skills and it follows it deterministically, like it knows how to fetch regions, which tool to use for it, how to predict splice-sites, which functions to employ to do this prediction. It knows from the skills where to get the API key and how to use it. So right now it will follow our instructions quite precisely and deterministically, but we don't need to put this into the code. We just, from our biological grounds, should think about what is an appropriate workflow to annotate genes, given the models we have, and push this into the prompt. While this is running, let's explore another application. So another task will be to interpret variants in promoters in humans. Probably those of you who are familiar with medical genetics knows that it's a huge problem for monogenic diseases that even although we have all these fantastic genome sequencing techniques, still the large portion, depending on the disease, could be up to the half of the patients with monogenic diseases cannot be molecularly diagnosed. It means that you have clear symptoms that indicate the heritable, probably monogenic disease. And after molecular tests, such as whole genome sequencing, you cannot identify direct cause of this disease. And of course, since you cannot identify cause, you can be sure in diagnosis, you cannot develop new drugs, you cannot select the drug, and you cannot design a reproductive strategy for the family. It's something we face a lot when we work together with healthcare providers. That's a huge problem in this area. And one example why this could happen, because those regions which do not encode proteins, and these are actually 98% approximately of human genome, we don't know functions of these regions. And when we see the variation in this region, we don't know how to interpret it. Is it the cause of disease, or is it just normal variation specific to this family? It could be quite rare, but still completely benign. That's the problem. And that's one application of genomic intelligence models that you can use. For example, there is a gene BMPR1A, which is known to cause a specific type of inherited predisposition, very strong inherited predisposition to cancers. It's known about the gene, but it's less known how this gene is controlled. And a couple of years ago, there was a publication about series of deletions in the sequence near the promoter of this gene. It was not really known what is the functional consequence of this deletion, because we don't know well the grammar of promoter. So we know if the promoter is completely removed, probably it will cause the disturbance of the gene, but we cannot interpret the variant identified in those patients. And as I said, why this is important, because approximately for half of the patients, we cannot diagnose this because from the whole genome sequences analysis, we cannot understand impact of this variant and other similar variants. So let's make an explorative study about what could be an impact of this deletion. How we do this? So we construct a prompt. In classical bioinformatics, we would construct ourselves the promoter sequence, we would do the deletion in silico, we would remove some bases, but let's do it in a agentic way. So we would say that the task is to fetch the promoter sequence for this gene from specific human version of human genome and prepare the standard file containing the sequence. We explain what is the region. We also can leave it, but this will be less deterministic. So I prefer to say specifically what we consider as a promoter. Then we again say that we would need to use skills and read instructions and follow instructions. And then we say that here is a sequence that is deleted from the promoter. So we ask the agent to delete this fragment from the sequence and prepare the file for original sequence and for the sequence with deleted fragment. Then we would need to, after we have these two files, the normal one called wild type and mutant, so we say that after this, we would need to determine how this 150 base per deletion affects the promoter activity. We say what are inputs, and we already said where are skills. Okay, let's start the execution of this one. It's loading now. Yeah, and it's going now. So it will explore the repo, it will find appropriate skills. Actually, it's nice. What I love about ClawBio, that for sure it's useful to combine skills from different sources. For example, here to get the DNA sequence, it will call for some other sets of skills, not genomic intelligence. Here it will call NCBI datasets to get the sequence. And that's very nice. So you don't need to think yourselves about each instrument that should be used in the workflow. You might specify the core set of instruments. You might leave even this to the agent, and it will work quite well combining different skills. And I don't know, Manuel probably will correct me, but I think there are several hundreds of skills now. So it's a very huge collection for all types of biology that can be
They are all
used.
random.
Random?
Random in the sense that is whatever people decide to contribute. Officially, I think we are about 80. So a long way to go.
Yeah, but plus this 80 that you have officially, there are many more from community.
Yeah, well, I mean, if you count then, because there are adapters to things like Galaxy, which then could make you have like 2000, but ones that have been pushed by people from the community are around 80 right now.
Oh, okay, I see. So 80 from community and then 2000 with Galaxy. Well, this is fantastic. So it's really a lot of skills. I don't know if we have actually many more, popular genomic instruments than this. So the coverage is quite good. And you see that the Codex, for example, can use it without explicitly prompting.
I
Okay,
stand
well, this
corrected.
one is
89.
okay. Yeah, cool. So while this one is running, okay, sorry, it's too fast. It's already done. So let's see what are the results. Yeah, so it says that original sequence without deletion is has a promoter activity, and the model is quite confident about this. But after deletion, the confidence drops below threshold, so it says that most likely it's not promoter anymore. And this is a very nice result because it matches the experimental validation, which shows that this 150 base pair deletion indeed abolished promoter activity. And we can read report here for details and ask some follow-up questions about like what could be consequence of this. For example, if this is a clinical genetics or someone not familiar with the gene, we can ask okay, what can be possible consequence, and then it will, based on the knowledge about the literature of the gene, it will say what would happen, and what can be the probable implication for the patient. Actually, especially useful note here that, of course, we all should understand that this is still in silico prediction. For sure, it should be validated in a laboratory, but it could save lots of time to select most promising candidates for laboratory validation because, of course, we have so many variants that we cannot validate every and each of them. We somehow need to prioritize. So that could be a good way to prioritize these regions. Okay, let me see if our previous task is also finished. Yeah, this one. It's still running. It's actually a longer one because it would need to do many different steps. And again, what is very nice here that it shows how to build workflow from very different instruments. So here it used two genomic intelligence models, one to find genes and their starts and their ends and the transcript itself, another one to find splice sites, but then, for example, BLAST will be executed using completely different set of skills. So you can embed models, predictive models, into workflows with the tools like this, with ClawBio skills. Okay, while this one is running, let's move on to the third task that I prepared. It's the last one. So here we would like to make an exercise on drug development. It's actually a very interesting study published just recently, a couple of months ago in Nature, that described that there could be a type of gene therapy for a cancer. Here, the therapeutic drug is a gene which encodes a toxic peptide that kills the cell, and also an immunomodulatory agent here. And what is very important about this drug, that the gene should be controlled by a specific promoter that will be active only in cancer cells, because the team, they target glioblastoma cells, and of course it means that they deliver this into the brain, and if this will be active everywhere, it will damage neurons, healthy neurons in the brain, and it will be too much collateral damage to use this drug. And to make this selective activity of the gene in cancer cells only, the team actually did a huge work by doing several experimental assays and then cycles of laboratory validations and improvement. And finally, it's a great success. They were able to design this sequence that can be active only in cancer cells. So the question is, can we speed up this process of development using this genomic intelligence model that can predict activity of the gene based on the sequence of its promoter? Can we, instead of running these cycles of experiments with incremental improvement, do this in silico using the model as experimental system, as a laboratory, predict the results, and go to the laboratory only with a small set of final candidates? That's something we want to try now. So for this, we will have a prompt explaining how it should work. So we need to design a promoter with a high predicted expression in glioblastoma stem-like cells and low expression or activity in normal brain. So we again ask to use ClawBio skills here to be precise, to read instructions and follow them. And now it's a good time to explain how we specify in the model that I showed on the web platform, on the model that can predict activity on the gene, how we specify the cell type, because here it's very important. The key for this drug is to make it active only in one cell type, but not in another. And what is, I think, very nice about these models that you specify it by a plain text. So you say that you will understand by activity a specific assay, this poly-A plus RNA-seq from adult human patient-derived glioblastoma stem-like cells. So the model can understand this description in human readable format. You explain like these cells typically express some markers, which we know are common for glioblastoma stem cells. And we say that we want to start with specific, so we did this assay using specific genome build, specific sequence design. So basically, since we want the model to reproduce the cycles of laboratory experiments, we need to explain, like, for example, like we explain to our students in a lab, what is the design of experiment in the same way we explain this to the model in a human readable format. And similarly, we should say about off-target context, like what cell types what are the cell types where we care not to have any expression of the toxic gene. We also recommend to start from the plausible endogenous human promoter, like from some sequence which is already in a human genome, and score different human genomic regions to find the best candidate. But once it's found, we ask the system to improve it in cycles of optimization, which means we want to change the sequence, score, instead of going to laboratory, score in the model how this improved the selectivity, the difference between target and off-target cell types. And then if the improvement is positive, keep this one and continue modification, like introduce one more modification on the top of it and another modification and so on. To make it a bit faster, we will do 100 cycles of this greedy optimization, but can be 500, 1000 cycles and even more. Okay, then we say what we want to see in the report. Like we want to see all candidates that were considered, what is the best candidate, how the process went. So we describe all this information. So you already saw it a couple of times, yeah? Again, with this prompt, we just put it into the Codex and submit. There will be a need to write lots of code. We will need some scripts to communicate with the model, to introduce these variants, to score them, to store results. All this code will be written by Codex, and it will follow the skills in a ClawBio repository to make this code communicate properly with the model. I want actually to highlight one thing here. So after reading the skill, it says that as an input, we need 9 KB single record file with the DNA letters centered around promoter. You remember when I showed you the models on a website, I mentioned that this is important to center the sequence in the gene start. So here, what's important, you don't need to think about it. We have this in skills, so it will be followed exactly in a correct way. Okay, and while this one is running, let's see what's going on with our gene annotation task. Okay, so it's still running the blast. We will come back to this later on. But we are almost in the end here, so it already identified some protein sequence here, and now it's trying to find what its function by comparing with other proteins in the data set, in the database. And what about our glioblastoma promoter? Yeah, this one is also running. I think that since we have not so much time, this probably would be done in 2-3 minutes, but maybe we can start the questions section. It was my last example, and while we have questions, I want to show these links again to the platform where you can explore models. This is my email. Please feel free to ask questions, to submit examples of how you use this CLOA with Genomic Intelligence. And please follow us if you are interested in updates in social networks. And I will keep this on screen, and maybe we can handle some questions now and then come back to the results of these agents. So if there are questions in a chat that are not answered, maybe Misha and Julia can read them, because I see there is a lot of discussion and I cannot evaluate what questions are not yet answered. So if you see some questions, please read them. And also, I think you can unmute yourself and ask questions aloud. The Zoom should allow this. And while this is going, let me check how the optimization goes. Yeah, you see, it actually already wrote all the scripts necessary for doing this optimization, and it's now submitting the API calls to the model, gradually improving the selectivity of the drug. So you can see there are already some variants. Oh, no, now right now it's scoring the endogenous human genes, like it's searching for the starting point toward development of this drug. And let's see what about this region. It actually says it's ready now. Cool. So what it found? It found some genes there. It found their structure with some splice donors and acceptors. It inferred a gene structure. It found some potential protein coding sequence there. It did search against the data set of known proteins, and it identified this one as a MYC gene. It's a proto-oncogene, very important for cancer development. Let's validate how it did work, because it made so many steps. I think it were like 8 or 10 steps there. So let's go to the genome browser and see, is it really correct that in this region located the MYC gene? Yeah, so it's nice. It's really reproducible. It found the gene which is indeed located in this region, and it identified it without any annotations, just by raw DNA sequence. Okay, I like this result. And what about the promoter optimization? Let me see if it's ready now. So it already found a starting point, and now it's introducing mutations into the DNA sequence to improve its specificity. Let's see how it goes. So most of variants are not accepted, but there are quite a lot that improve the sequence. So it's now about halfway done. We required 100 cycles of optimization, so should be done soon. And it started from something like 2.5. It's a difference in a log scale between like on-target cancer cells and neurons that we don't want to damage, which is 250 times, already good enough. But now it already improved it to almost 300 times by introducing these mutations. Yeah, I think I see there is a hand raised. Please feel free to ask. Yeah.
We cannot hear you.
Wyatt, we cannot hear you.
You can also type the question in a chat, maybe.
And there is another hand. Please unmute yourself and please ask the question.
Hi there. This is really cool. My question, I have a lot of experience using a different repo called Paperclip that has kind of the same deterministic ethos. They're really, really good at searching whole text of papers, making them sort of Git-like, where you can commit repositories, and like I can kind of see the obvious parallels here, where you would want to do some literature informed sort of background, then you would want to like pull your candidates through some sort of pipeline. Like, can you use agents from two different repos to sort of mix and match and kind of use your agent as an orchestrator across lots of these different kinds of skill repos?
Manuel, do you want to answer this one, because I think it's more related to how ClawBio is organized?
So at the moment, that's something that it doesn't happen. You have ClawBio where it has its own orchestrator. So inside it's able to sort of say, use this skill or that other skill. But I think it would be interesting to expand that functionality so that other types of skill repositories are compatible. The issue is that we have the compatibility aspect where, in a way, ClawBio is trying to have a standard in terms of how biodomain skills should be or would be defined in terms of a specific set of requirements, and we have no standard on that. So I mean, something like an MCP standard for skills might be useful here. So the answer is no, we don't have that capability, but it's something that certainly is where the frontier is. Sorry, I cannot answer better this question.
But I mean, it seems easy enough to build. Like I'm playing around with my own Claude code agent as we're talking because I have it installed. Like as soon as I can get a virtual environment with both repos basically going, I don't know. I want to play with this and see if I can build something in the next couple weeks because it seems like all of these are really interesting kind of computational experiment type skills. You're like running an analysis, you're trying to find a gene promoter, you're trying to predict a therapeutic index based on some model. If you could connect that to sources of knowledge in a way that was structured, like you're talking about, that has this really deterministic ethos, that you're not sort of hallucinating the random names or the random IDs, it could be quite powerful.
Yeah, I mean, again, I think you said it better than I could. It's completely research at the moment. Guys, I think we probably need to finish now because this was supposed to be 90 minutes. Any final thing you'd like to say?
Yeah, let's spend just like 10 seconds in seeing the final results of this final test. It's ready now, so that you have the feeling that every task is finished. And here we see actually that the system was able to optimize promoter by increasing, by first, it was able to find a suitable starting candidate quite well. And then it introduced about 30 mutations that improved this candidate by approximately like 20 times. Yeah, and that shows that you really can generate these candidates for lab validation in a couple of minutes. This was a remark about this specific task, and in general, I think we showed just few examples. There are many more, so please feel free to use the models. I think it's very easy now to integrate them with the ClawBio, and here is again some information that can help to integrate this into your research workflows. And
So
thank you, Manuel, very much.
Thank you, guys. We're gonna leave it there. There's a little, small, tiny, wonderful favor I'd like to ask you. If you wouldn't mind just starring ClawBio, if that should be something that by any chance you found this useful, please star it, because it's not a vanity measure. It helps. It helps. Everything, as you see, is for free. And yes, I will follow up. We will follow up with all of the links, with all of the information, with the recordings, with the presentations, and with everything. So I'm going to stop the